So in cancer imaging, as in other medical imaging applications, a lot is about knowledge discovery. We do not understand all the mechanisms of cancer. We do know bits and pieces. We can, if we analyze cancer images, for example, we measure the size of the tumor, the volume of the tumor. We try to understand maybe what makes it behave differently if put under a certain drug regime or a certain therapeutic regime. And similarly, the other two colleagues who spoke there, they work more on the genetic profiling and the genomics aspects. And what I'm doing used to be what's called radiomics, but we actually all use similar tools, even though we're working on completely different types of data. And neural networks, they're quite agnostic to what data you put in. They allow us to make sense of the data, to uncover hidden trends which we can't see because there's just too much data. We can't see the tree from the forest, basically, or the forest from the trees, the other way around. So these neural networks, if we train them on enough data with some annotations, we can actually all of a sudden see things which we couldn't before. Also, machine learning generally we apply in cases when it's just too labor intensive or maybe even impossible to manually process the data. One example I've given, for example, is extracting the tumor microvasculature of multiphoton microscopy. I mean, this is I've forgotten the number. I think it's 13 billion pixels a single data set has. So in principle, an expert would need to go through and label each individual pixel as to being vasculature or not, and that's just not possible. So annotating a small subset of that, training a network and applying it to the rest of the data actually helps solve these problems.